So welcome back friends. In this video tutorial we will going to talk about on the regulation of the complement pathway. So regulation is necessary for any kind of thing in this universe. Okay, you there are various regulation available. Let's say if we are talking about any kind of enzymatic reaction, there will be obviously feedback mechanisms available to control that reaction, right? That means control is necessary for any kind of thing, not only in biological systems. If we try to relate this with our day-to-day life, everything is necessary to be controlled, right? Because now let's say I used to play games for a lot of time, right? And that is why my career was in doldrums for some month. Okay, and later I have realized that why it is going on, and then I try to control it, right? Similarly, there are everything. If you talk about everything, you may find the control, right? Control is necessary for any kind of thing, right? If you have a horrible eating habit, then you have to control it. Okay, similarly, in case of complement pathways that we have just completed our discussion, in complement pathway, let's say for classical pathway, alternative pathway, and your what your lectin pathway, there should be some regulation that will prevent or that will protect our self cell from those complement proteins, right? Because you know what, if you study them very carefully, then you can understand that classical pathway or lectin pathway or alternative pathway cannot recognize what they are doing, right? And that is why classical pathway can be activated by various type of things. Lectin pathway can be activated by any kind of thing that have terminal mannose residue. Similarly, your alternative pathway can be activated by any kind of things, right? And if by chancely our self protein or our self cell contains those kind of composition, then they are at risk from those kind of complement proteins, right? And as a result, our immune system also develop some complement regulatory proteins which are present at high concentration in normal serum, and their main goal is to protect the self cell from the action of complement proteins. Clear? So now if you focus on the classical pathway or your alternative pathway or your lectin pathway, if you somehow prevent the formation of C3b or regulate the amount of the C3b, then your job is done. Because if C3b is not formed, C5 convertase will not form, right? And if C5 convertase will not form, there will be no formation of C5b. And if C5b is not formed, the terminal pathway cannot be start, right? And that is the idea. So most of the complement regulatory protein focus on to prevent the formation of or to regulate the formation of C3b. Okay, so first type of protein is C1 inhibitor. Okay, so this C1 inhibitor will going to bind an inactive C1R and C1S. So remember, in the classical complement pathway we have discussed that C1R and C1S are serine protease. Okay, without the presence of C1R and C1S, C4 and C2 component cannot be activated, and as a result C3 convertase cannot be formed. Okay, and if C3 convertase cannot be formed, C3b cannot be produced. Okay, and that is how C1 inhibitor works. So if you have not watched the video on classical complement pathway, then I would like to recommend you to please go and watch that video. So now let's talk about another regulatory protein, that is protein H. Okay, so this protein H will specially work in case of alternative pathway, remember it. Okay, and remember in alternative pathway, I have told you that the first step is the spontaneous hydrolysis of C3b, spontaneous hydrolysis of C3 and the formation of C3b, and this C3b is membrane bound. Okay, so here the protein H will going to prevent the binding of factor B to this C3b. Okay, so although here now you can understand this, that it is C3b. So the role of factor H is to prevent the binding of B to this C3b. Okay, and we have discussed that if B cannot bind with the membrane bound C3b, it cannot be cleaved by the factor D, and as a result formation of B small b cannot occur. Okay, and as a result C3 convertase cannot be formed, and which inhibits the formation of C3b. Clear? So C1 inhibitor works by inactivating C1r and C1s. Protein H will going to work by preventing the binding of factor B to membrane bound C3b. Right? Now let's move. So now another one, that is factor I. How they work? Now let's say somehow C3b is formed despite the presence of your C1 inhibitors and your protein H. Okay, now this factor I can further cleave this C3b into two small fragments named C3c and C3d. Okay, which are usually inactive. Okay, now remember it, this factor I can only work on cell membrane bound C3b, mostly on red blood cell, and that is obvious. If the complement pathway, especially the alternative pathway, start to act on red blood cell, it will be very dangerous for us, and that is why factor I present there to convert the C3b into C3c and C3d. Okay, so that the alternative pathway, mainly the terminal pathway, cannot be activated against red blood cell. Okay, so first we have discussed about the C1 inhibitor, whose main goal is to inhibit C1r and C1s. Then we have discussed about your protein H. The main goal of protein H is nothing but the prevention of formation of B small b by preventing the binding of factor B to membrane bound C3b. Factor I will going to convert the cell membrane bound C3b into C3c and C3d, which are inactive. Okay, and as a result it cannot facilitate the formation of C5 convertase. Okay, now the last one, that is S protein. This S protein will going to play the role of inhibitor, especially in the terminal pathway. Okay, remember at the end of the terminal pathway, I have told you after the lysis of the cell, the C5b67 complex can release. Okay, now this released C5b67 can bind with further nearby cells. Okay, and there is a possibility that this C5b67 can bind with self cell. Okay, and to prevent this kind of horrible situation, because if C5b67 can bind with the self cell and insert into the cell membrane, then C9 will bind, because I have told you C5b67 is the receptor for C9, and then it will facilitate the formation of poly C9, and as a result our self cell can be lysed. Okay, that is why S protein presents there. This S protein will bind with C5b67 and facilitates a hydrophilic transition. Okay, this hydrophilic transition will prevent the insertion of C5b67 into the membrane of the self cell. Okay, and as a result it cannot create any kind of problem. Okay, so that is the fourth type of regulatory proteins present that protects our self cell from complement protein. First one is the C1 inhibitor that will inactivate the C1r and C1s. As a result, C4 and C2 cannot be activated. Second one is the factor H, or let's say protein H. This protein H will going to prevent the binding of factor B to membrane bound C3b, and as a result the factor B cannot be cleaved into B small b, which ultimately prevents the formation of C3 convertase. Now the third one is factor I. This factor I will convert the cell membrane bound C3b into C3c and C3d. Those are usually inactive, okay, and as a result they cannot facilitate the formation of C5 convertase. Now the last one is S protein. How they work? Once the membrane attack complex completed its action on the foreign cell, the released C5b67 can bind with nearby cells, that may be a self cell, and inserts into the cell membrane. Okay, so the goal of S protein is to prevent this insertion. How they do this? They do this by binding with the C5b67 complex and by facilitating a hydrophilic transition. Okay, and this hydrophilic transition will prevent the insertion of C5b67 on the cell membrane of a self cell. Okay, and as a result the self cell can be protected. Clear? So that was the idea about the regulatory proteins. So I hope this video will be helpful for you. Thank you. So now we have just one video left, that will be on the biological consequences of complement proteins or the complement pathways. Then we will going to solve some MCQ question from that, and that will be the last video of this playlist. So I hope you are enjoying this playlist. So thank you for listening to this class.